Great. Thank you very much for that introduction. It's great to be here this afternoon. Good afternoon to everyone. So yes, my background, it's been very varied with my postdocs, as are detailed there. I've been all over the UK and a few postdocs in the Middle East as well. The majority of my research is in the Middle East on the Neolithic, but recently I've been branching out and working on some different projects in the Caribbean and South America as well. So today's talk is very much a methodological talk. I'm going to discuss how we can integrate different methodologies to try and identify animal management and domestication. So I wanted to start by putting this into a bit of context. So looking at why it's important to integrate these different methods. So I just wanted to start with this quote from one of Dorian Fuller's papers. So this says that the transition to farm is often regarded as the most significant threshold in recent human cultural evolution. So in a nutshell, what Dorian's saying in this quote here is that agriculture is as a prerequisite for civilization. So it's a really important subject that archaeologists are studying worldwide, but today I'm going to focus on some of my PhD research and results in the Middle East. So looking at farming, as I said, is very important. It's sort of the thing that changed everything and led to where we are today. So just to look at a few stats here, you've got pre-industrial revolution, about 7% of the land was given over to agriculture. And then today, about 40% of the land is now used for agriculture. So you can see that big changes in agriculture have led to where we've got to today. So really looking at this transition is very key, and for decades and a long time everyone's been trying to look for evidence and different ways of looking at the evidence to try and identify this big shift. So trying to find out when people shifted from this mobile lifestyle, hunting the wild animals, gathering the wild foods, and becoming more sedentary, living in villages, managing initially, and then domesticating their plants and animals. So it's a big change, and in the past, initially people referred to it as the Neolithic Revolution, but in actual fact we know now from much archaeological research that it happened over a very long time period, different centres of domestication, different domesticates as well. So really looking at this transition is really key to understanding the origins of modern human society, and that's the context of what I'm going to be discussing today. So the domestication of plants and animals, we now know that there's multiple domesticates and domestication centres worldwide, from the domestication in the New World in the Americas to the Old World domesticates and domestication in the Middle East, Asia and Africa. So while very different fauna and flora in different parts of the world, and also happening at very different times. So today I'm focusing on some case studies from the Middle East. So this is very much seen as the first farmers worldwide, so the earliest domestication about 12 to 10,000 years ago. Today I'm going to be focusing on the domestication of animals, management and domestication of animals, but this comes as sort of a package within a range of plants as well. So you've got different types of wheat and barley, legumes, and then with the animals you're looking at the sheep, goat, pig, and cattle mainly. So the case studies that I'm going to focus on today are the sheep, goat and pig. So I'm going to show you a couple of case studies from research I did as part of my PhD project. So you can see from this figure here that the sort of earliest domestication is about 11,000 BP. So traditionally, people look to archaeozoological approaches for identifying management of herds and domestication of herds. So traditionally, archaeozoologists would look at the bones to look at the size and shape, so looking for changes in overall body size and changes in the horn size and shape as well, which is a result of the domestication process. And looking at the bones, things have moved forward now, and people tend to look at herd structures and kill-off patterns rather than just this morphological change in sizes from the animal bones, because as research has progressed in the archaeozoological research, it's now sort of accepted that the morphological responses and the change in body size happens later on in the domestication process. So if I go back to this graph here, you see 11,000 is sort of the earliest date on here, but it's not until about 9,500 to 9,000 that you see the body change sizes, the morphological changes. So as I said, the archaeozoological research has moved on using a lot of geometric morphometrics to start looking at herd structure and culling patterns in the archaeozoological remains. So it is by looking at the bones and doing various measurements that people look at identifying the sex and the age of the animals that are in the assemblages. And typically a hunting archaeological site has very large adult male bones. So the large male animals are being hunted, so they have obviously the most meat on them. But when you have an archaeological site which is more representative of farmers, early management, domestication, sedentary villages, what we see instead is a dominance of two types of animal bones. So you get very young male animal bones and very old female animal bones. So they're killing off the older females and the younger males. They're really keeping the females that are able to reproduce. So they're managing the herd in order to grow the herd size, and they're killing off the animals that they don't need for reproduction. So whether they've got too old to reproduce or the young males, which they don't need so many of. So this is a sort of analysis, obviously, which people moved on to rather than looking at the morphological body size change. The other way that people were traditionally looking at was the horn cores in the animals, looking at the size and shape, and this does change as a result of domestication, and it's definitely a change because of human influence, but rather than what the influence is with the change in the horn cores is where the animals are being moved. So the anthropogenic influence is moving the animals outside their natural habitat into a different environmental area, and this is why the horn cores change. This is another thing that archaeozoologists obviously look at if they have it preserved. So looking at this figure again, the coloured shaded areas show the earliest evidence for these different domesticates. So it's in the eastern fertile, northern fertile crescent, and then the coloured numbers in the other regions show at what date that these domesticates, these animals were being domesticated in other regions of the fertile crescent. So in the eastern area, it's sort of 500 to 1,000 years earlier, and that's where I'm going to be focusing the results I'm going to show you today. So why was it first in the Zagros rather than the Levant or the northern fertile crescent? So this is an area, the Zagros Mountains, where they have very diverse topography and resources, and all of the wild species that were brought under domestication live in this area naturally, and so it's an area where they lived they were wild and they lived naturally, and also these were species which were suitable for domestication as well. So over in the Levant, in the western fertile crescent, the natural species are more, for example, like the gazelle. So looking at the gazelle, they are behaviourally unsuited to domestication. They're very territorial, they're very nervous. So while some academics have argued that there's evidence for gazelle population management, it's generally accepted that the eastern fertile crescent has the earliest domestication with the sheep and goat, where they were living naturally in that environment and brought under domestication. So traditionally, that's how our specialists on archaeological sites are trying to look at this process of management and domestication. So traditionally, they're taking all the bones, they're measuring them, they're looking at size change, horn core change, they're looking at the herd profiling, the kill-off patterns. And if you have lots and lots of animal bones, like a couple of these pictures that I put up here, you can get all the measurements. You can work out, are they male, female, what age they are, and all the detail that you might need to make your interpretation. But the evidence that I'm going to bring in really comes into play and really helps to support the interpretation of management and domestication, is if you don't have very many animal bones, or if the bones are poorly preserved. So obviously, to do all of the measurements, the geometric morphometrics, you need a well-preserved bone, hopefully removed from the ground intact, and so you can do all these measurements. So here I've shown 3 bones from some of the archaeological sites in the Zagros that I worked on as part of my PhD. So the tibia, scapula and femur there, you can see they're very degraded, they're very broken. They've even been measured to some degree prior to excavation because they're breaking up. So if you don't have a good preservation of animal bones, or they are completely dissolved within the archaeological deposits, perhaps due to these the soil conditions or the age of them, for example, it can be very difficult to do some of these analyses. So here I've just shown, this is some fragments of poorly preserved bones from some of the archaeological sites. They're very broken down, very fragmented, so often they can't even be identified to species, let alone their age, their sex, etc. So bones which would be able to have nice measurements like these ones on the left, if they're not intact, it's very difficult for the archaeozoologist to then identify the status as whether they're wild or domesticated. So this is where I wanted to introduce some of the research that I've been doing on identifying ancient animal dung. So this is using more non-visual microscopic signatures to try and aid this identification of management and domestication. So before I explain really how that can be done, I wanted just to go over a little bit about how we can detect ancient animal dung in our archaeological sediments. So one of the key indicators of animal dung is the presence of what are referred to as fecal spherulites. So these are very small, 5 to 15 microns. They're composed of calcium carbonate, and they form in the animal's gut during digestion. And when you have some sediment from your archaeological site and pop it under a microscope in cross-polarized light, you can see these fecal spherulites because they have this very distinct cross of extinction. So they look like little hot cross buns. So I've got a picture of highlighted a range of them here. They can be mistaken for starch, and starch can be mistaken for fecal spherulites because they have a similar cross of extinction, as well as a few other particles such as coccoliths as well. But people that specialize in animal dung look at the bifringent properties or the chemistry of them as well to distinguish them between these other microparticles which have the same cross. But if we do identify these fecal spherulites, so they preserve very well through the animal's digestive system and preserve in the dung. So if we find these fecal spherulites, they're a key indicator that animal dung is present. So once we identify the dung, how can it actually help with identifying early management and domestication? So if the animals are morphologically wild but being put in an animal pen, this is obviously an indication of some anthropogenic influence there. So even if the humans are putting wild animals in a pen, it's showing the early stages of management. So if you find the animal pens, regardless to the evidence from the animal bones, whether it looks like a wild herd, domesticated herd, something on the cusp, or you don't have very many bones or well-preserved bones, the evidence of an animal pen is at least evidence of management, if not domestication. So it's finding these fecal spherulites and these dung deposits in a specific context, and the way that we can do that is using what's called micromorphological thin sections. And I'll go into a little bit more detail about how we identify different types of dung in thin section. But when the animals are put in the animal pens, they're obviously compacting the dung, so their weight means that all of the dung becomes compacted, laminated, and it's very distinctive under under the microscope. And while this is a thin section from a modern dung pen, modern animal pens, which have put this little excavation in Iraqi Kurdistan, all of this top layer here is animal dung. In modern samples, you can see the organics, the vegetation, but over time that will degrade. But what you'll end up with is silica phytoliths, which still preserve and still represent these compacted and laminated dung deposits. So the sort of thing that we would be interpreting if you find this these deposits would be something like this. This is a modern traditional village in Iran, where they're using a part of their outdoor area as an animal pen. So a lot of modern studies and reference collections are now emerging so that we can study animal dung in a known context to compare to our archaeological deposits. So as well as the penning deposits, which we look at in these micromorphological thin sections, you can identify these fecal spherulites in thin section, but also very quickly under the microscope, you can pop a bit of the soil with something like glycerol or clove oil. So in the field, if you've got a microscope, you can identify the presence of dung without having to take a micromorph sample and take it home to the lab. Another thing that can be analysed from the dung is these silica phytoliths, so they preserve very well as well through the digestive system. So you can start to look at things like animal diet as well. So the first thing that I applied during my PhD in order to try and identify dung deposits was portable X-ray fluorescence. So looking for elevated phosphorus on archaeological sites. So obviously the things we're looking for are mainly non-visual, so you can't often see these dung residues, these dung deposits, because until you put them under the microscope, you would not be able to see the presence of dung. But what we did use in the field was this portable geochemical X-ray fluorescence, and elevated phosphorus can be a key indicator of animal dung. So if animal dung is deposited in and around an archaeological site, it will raise the level of phosphorus there. Although just to point out, it's not the only thing that raises phosphorus. So it may be dung, but also may be indicative of other activities. So your hearths and your fires, for example, those sorts of deposits will raise the phosphorus as well. Any sort of bone input into the deposits or food residues also raise the phosphorus. So the approach that I took during my PhD was to use the PXRF to find the phosphorus, and then take small amounts of soil, pop it under the microscope, and see if I could find any of the fecal spherulites to rule out some of the other sort of inputs. So once I found a location which seemed that it would probably have some dung based on the geochemistry and these smear slides to look for the fecal spherulites, I cut micromorph blocks, so cutting a block into your targeted location, wrapping it and taking it back to impregnate with resin, and cutting and polishing it and grinding it onto a large format thin section. And then this can then be put under the microscope to try and look for the penning deposits. As I mentioned, as a part of my research, I've been doing a lot of ethnoarchaeology and modern reference materials. So to look at different types of dung microscopically to see how we can distinguish between different types of dung under the microscope. So during my PhD, we did a small ethnoarchaeological investigation in Iraqi Kurdistan, and since then, as part of my current postdoc, I've been looking at a much wider signature of different types of animal dung, comparing different species, wild and domesticated, and looking a lot at diet as well and seasonality to see if we can identify animals in various locations at different times of the year based on what they're eating. But typically under the microscope, herbivore dung in thin section is very brown, and you get a lot of these fecal spherulites and a lot of silica phytoliths. And the phytoliths mainly usually come from grasses. You can identify cereals such as wheat and barley in it as well. And often the fecal spherulites, which I've circled in this bottom picture here, are sort of adhered to the silica phytoliths as well. So you find them very much sort of overlying the silica phytoliths. So and then comparatively, if you look at omnivore dung under the microscope, you've got a lot less of these phytoliths and a lot less of the spherulites as well. So herbivores produce a lot of these fecal spherulites and omnivores produce a lot less. And because the diet of omnivores such as pig or wild boar is much more varied, you get lesser input of plants and input of other materials. So they do a lot more sort of foraging and rooting around, eating roots and berries and fruits. And also a lot of examples have shown small fragments of bone in the omnivore dung as well. So there's less plant material, less phytoliths, less spherulites, and more orangey in appearance. So you can start to distinguish between types microscopically using comparative material. So the project that I'm going to give you a couple of examples of how you can integrate this fecal studies with archaeozoology comes from the Central Zagros Archaeological project. So this is a University of Reading project run by Roger Matthews and Wendy Matthews. The project excavated 4 different sites, 2 in the Iranian Zagros, that's Sheikh-e and Jani, and 2 in the Iraqi Kurdish Zagros, so Bestancer and Shimshara. And the dates are roughly from 12,000 to 9,000 BP. So today I'm just going to show some example from 2 of the sites, so Bestancer and Sheikh-e. And I've chosen these 2 because they are representative of different types of animals. So in Bestancer, this is in Iraqi Kurdistan. It's in the lower Zagros Mountains, about 550 meters above sea level. This is a tall tell site, 7.5 meters high, and the Neolithic occupation here, while it is underneath the mound itself, it also extends to the fields around the mound. So in total, it's about 5 hectares in size, this site. So it's quite a big site, and we put in 13 trenches into the side of the mound and also out in the field surrounding it. Fortunately, we couldn't get to the deposits right underneath the center of the mound, so the Neolithic occupation is overlain by Iron Age occupation. So unfortunately, while we did some trenches into the edge of the mound, we're unable to get right to the center of the village. So the archaeozoological results from Bestancer represented in this graph here in the black, dominated by sheep, goat and pig. So those are the 3 dominant species here. And this is the work of Robin Bendry, who's at the University of Edinburgh now. So I'm going to focus on the evidence, the archaeozoological evidence for wild boar or pig, and then I'm going to show you some evidence for from the fecal material to try and back up or support the evidence for the archaeozoological remains. So obviously, being omnivores, the wild boar or pig have, as I said, a much wider diet. So while they will eat plants and vegetation, they pretty much will eat anything as well. So the pigs are, the wild boar pigs are attracted to human waste on early Neolithic sites. So where the populations are becoming more sedentary, they're staying in one place, they're having a lot of waste material. We see on a lot of Neolithic sites large areas of middens where they're dumping all of their waste. So while wild boar may have been hunted, so they prey and like the people go out and hunt them, you also see here with these animals being omnivores a more commensal relationship as well. So they're attracted by the humans, and then the humans are going out hunting them as well. So the earliest human management precedes the morphological changes, so it can be very difficult to identify these sort of early management and domestication evidence. So the earliest pigs are actually not that morphologically not that different in size to their wild counterparts. So as detailed earlier, this can obviously take up to about a millennia. So at Bestancer, what's really interesting is the percentage of the overall assemblage which actually comes from wild boar or pig. So this graph here shows a range of Neolithic sites in the area, and mostly the percentage of wild boar or pig is below 10% for most of these sites. But at Bestansur and another CZAP site of Shimshara, you actually see a very large amount of wild-born pig here. So at Bestansur, it's nearly 30%. At Shimshara, it's up to as high as 60%. So the number of the percentage of animal bones which are from wild boar or pig suggests a major dietary value at Bestansur. So looking at the bone results, looking at the demography, we would expect that if they were being managed and domesticated, that they would be slaughtered when they reach full body size. So it's about 2 years of age. So if this was a managed and domesticated herd at Bestansur, you would expect to find a lot of animal bones from up to 2 years, but not that much older. However, looking at the results of the bones that were able to be measured and identified to age, sex and age, the majority of them are actually culled older than 2 years. So of the ones that can be identified, you've got 9 which are culled over 2 years and only 4 which are culled under 2 years. So this is suggesting from the demography, older animals, which would suggest wild rather than managed or domesticated. However, then if you look at the size of the molars, the range, the size and range at Bestansur is very similar to a nearby site of Jarmo and the pottery Neolithic levels, which are interpreted as a domesticated herd. Bestansur has a similar size range, but also has some teeth which are even smaller than at Jarmo. So while the demography is suggesting older animals, the tooth size is suggesting management or potential domestication. So this is where bringing in alternate lines of evidence can start to help pick apart these interpretations. So at Bestansur, I found a lot of herbivore dung, but no evidence from the herbivores of penning. Not that the pens weren't there. As I said, we can excavate under the main part of the mound, so it might be just that we haven't found any herbivore pens. All of the ash material from the hearths and fires at Bestansur all had animal dung being used as a fuel, which is again another indirect suggestion that of management or domestication. But when it comes to the omnivore fecal material, I'm going to show you some evidence from trench 12 and 13 at the north end of the site. So you may or may not notice at this point I'm referring to it as fecal material rather than dung, and I'm going to come back to why that is in a moment. So this trench was cut into the edge of the mound, and this is the area. So not in the centre, but just right at the edge of the occupation. So in this area of the site in trench 12 and 13, you could see some slight orangey sort of layers showing up during excavation and cleaning of the section. Sort of lots of repetitive dumping activities and also repetitive what looked like potential surfaces. These were examined for the geochemistry in the field. They had very, very, very high levels of phosphorus, and conducting some smear slides of these orangey layers. I was also finding some fecal spherolites. So these thin sections were taken, and over about 1/2 meter, there was eight different layers of fecal material. They range between 1 and 6 millimeters in total, and under the microscope you can see it's very orangey in color, sort of compacted, laminated and repetitive. There was hiatuses between the different layers, but repetitive layers of omnivore fecal material. Within the fecal material that was identified in this area, there's lots of small fragments of bone. So some of it was fish bone, some of it probably bits of microfauna and other types of bone. So you can see it embedded within in the dung here, and it's showing up in all of the layers. So you have these sort of low numbers of fecal spherulites. You've got the bone, it's very orange. There is some phytoliths, there is some plant material there, but it's quite low. So the reason why I said it's omnivore fecal material is that another method which I employed during my PhD was GCMS, gas chromatography mass spectrometry. So this can, by looking at the bile acids and coprostanols, you can distinguish between herbivore, pig and human. So you can't distinguish between sheep, goat, but you can distinguish whether it's omnivore or herbivore, and then with the omnivores, whether it's derived from pig or human. So of the samples that I tested in this section, all the lower layers actually were human in origin. So rather than saying it's omnivore dung, that's why I was saying it's omnivore fecal material. But the uppermost layer came back via the GCMS results as being wild boar or pig. So what you have here is repetitive areas of either like a waste area or a human latrine, and then finally you have the pigs coming in, and the layers are laminated and compacted. So it's pigs or wild boar coming into this area of the site and congregating or being corralled there for a period of time. So pulling all the results together, you have a lot of pigs at the site, and then you have this opposite information from the age of the pigs and the size of the teeth, so older pigs and smaller teeth. But we definitely do have a layer of pig dung in an area where there has been a lot of human waste. So the animals are being attracted to that area perhaps, and either captured and penned or congregating in that area. It does show that the animals are there on site. They're not being hunted off site and brought in. So the evidence from the dung studies can contribute towards trying to get a better picture of the management and domestication. So I'm going to move on to show the results from one of the Iranian sites, Sheikh-e. So I want to show this example because rather than being evidence for omnivores, it's providing more evidence for herbivore management and domestication. So this is in the upper Zagros, 1,400 meters above sea level. Again, it's a tell site, 8 to 10 meters in height. 3 trenches were put in on this site from the earliest to the latest occupation. They've got no more occupation over the top of the mound, so it's straight below the plough soil into the Neolithic levels, dating from about 11,800 to 9,600 BP. So looking at the archaeozoological results, there is poor to fair condition of the animal bones recovered here. So from one excavation season, there was about 2,000 fragments of bone recovered, but only 200 of these were actually diagnostics. And of that 200, half of them were sheep and goat. And this is a site that had no pig remains recovered. It's not to say that wasn't pig there, but in the faunal remains with the preservation, none were identified. So mainly it's the sheep goat, but not great preservation here of bones. Trenches 1 and 2, so the earlier trenches, the poor preservation, so the domestication status is unknown. In trench 3, some of the sheep and goats identified were allocated as managed or domesticated. What they also found at Sheikh-e was this ritual deposit of 5 animal skulls. So it was a T-shaped rectilinear room, and it has 4 goat skulls and 1 sheep skull deposited. So the tops of the horns have actually been broken off from the plough, because this top trench is very close to the plough soil. This shows a reconstruction of what they thought it might have looked like. So 4 of the skulls, the 4 at the front, were goats, and they were wild male goats. So while in that trench at the top there, we do have some indications for management and domestication, this deposit specifically, morphologically, they are wild. So, but as I've already sort of mentioned, you can have long periods of management and domestication of animals before morphological changes come in. So these animals could be at the early stages of domestication, but morphologically they look wild. So looking beyond the archaeozoological results, I'm going to show you this one example of an area where some penning deposits were identified. So this is that ritual deposit in this T-shaped room. You can see the skulls there. So these are morphologically wild sheep and goat. And then in building 1 in space 8, during excavation, there's about ½ meter of repetitive layers again, and in the field it actually looked like these were potentially plastered floors. These were sampled, block sampled for micromorphology, impregnated, and looked at under the microscope. So you can see here the slight color change, repetitive layers of browny orange material. And under the microscope, rather than being plastered floor layers or trampled floor layers, they're repetitive layers of compacted and laminated herbivore dung. So these have lots of these fecal spherolites, lots of phytoliths from grasses and cereals identified. So this is an area which has been repetitively occupied by animals. So it may have looked something like this modern example that I showed earlier. So an area where animals are taken on and off, they're put in there, there's hiatuses in between, and I'm not going to present it today, but the phytolith results from the different layers shows some indications of seasonal occupation as well. So we can look at when these pens might have been identified by looking at the plant remains as well. So in Sheikh-e Bad, there was a lot of sheep and goat in comparison to Bestansur. The domestication status is unclear from the archaeological remains. So with the poor preservation of diagnostic bones, it becomes difficult to come to a firm conclusion. Definitely that ritual deposit is morphologically wild, but whether they're at the early stages of management and domestication, that can't be ascertained. But what it does show is that using the an alternate approach by looking at the animal dung and identifying deposits under the microscope, at least management, if not domestication, is identified from the animal dung using the microscopic remains. So it shows how that you can really integrate these methods to try and ascertain the management and domestication status of herds, different Neolithic sites. So, so just to conclude, hopefully I've showed how you can use new types and different types of evidence and integrating methods well to be able to get a better picture of management and domestication. And I didn't go through all of the methods, but there's lots of different methods you can apply to dung studies, and often it's things that we can't see visually. So it's this sort of microscopic clues from the sediments, which can actually tell us a lot of information beyond the macroscopic remains that we get from our sites and that the archaeozoologists examine. And more and more projects are now looking at dung studies and using animal dung, and it's something which is really expanding, and all the different aspects of dung studies can answer a wide range of research questions beyond management and domestication, because we can look at things like secondary product use, fuel and fires, sites that aren't very forested at the time of occupation. We can see how dung, for example, is being used in construction and also in as a fuel as well. Thank you for listening.